Liftoff. We have a liftoff.
It's been compared to landing a person on the moon.
Liftoff on Apollo 11.
But when Neil Armstrong took that first step,
That's one small step for man.
there was nothing ambiguous about it. History had been made. Compare that moment to this week's announcement, the first draft of the human genome, the map of all our genes. We're told the discovery is monumental.
Without a doubt, this is the most important, most wondrous map ever produced by humankind.
But just what is the human genome anyway? You've heard of DNA. In almost every cell, those spiraling chemical ladders, on the rungs, tiny commands that when taken together hold the complete instructions, the genome for making a human being. Enough information to fill 200 city phone books, billions of letters of instruction. So far, scientists have decoded 90%.
We have some work to do to clean this up and get it into the final finished form, but the vast majority of that information is there for everybody to look at, study, and try to figure out what it means.
To decode the genome, scientists fed bits of human DNA into machines that actually pick apart and read the letters that make up the body's commands. There are only 4 letters in all, A, C, T, and G. Create different combinations and you've spelled out the body's genes, which control the color of our eyes, the texture of our hair, and even the type of diseases we get. Researchers think there are 50,000 genes in all. Trouble is, right now they can't decipher most of them.
The information in the human genome is basically a parts list. We now have the parts, but it's going to be the work of the next decades ahead to figure out how those parts get put together to make a human being, and then how a human being really works.
Compare that to early anatomy charts. They didn't tell doctors how to fix, say, the heart or the lungs, but did at least reveal the location of those organs. Same for the human genome. No solutions, but invaluable clues. One payoff, with a map to guide them. Researchers can now find genes in just weeks or months instead of years.
More than a dozen genes involved in important human diseases have been discovered. They would not be discovered yet if we didn't have this information.
Another potential benefit, more accurate diagnosis for patients. Already genes are revealing important information about diseases like cancer. For instance, tissue from 2 lymphoma patients looks almost identical under the microscope. But now look at their genes. The genes reveal the patient on the left has a 75% chance of being cured. The patient on the right, less than 25%.
They are reading the instructions from the human genome differently. That makes the cancer cells function differently and respond differently to our therapy.
But perhaps the most revolutionary change will come in the treatment of disease, tailor-made drugs, and even better, fixing the defective gene. It's been done once already, earlier this year, in what's being called the first successful cure in gene therapy. To treat patients like this child suffering from a life-threatening immune disorder, commonly known as bubble boy disease, French researchers replaced the defective gene with a normal gene. They treated 2 children. Months later, they're still healthy. The genome map promises the discovery of other genes that doctors may one day be able to repair.
It's a critical set of instructions. All of the ways that we approach disease, all of the research that we do will be different from here on out.
And if that's not enough, biologists can't wait to mine the genome for clues about the human family tree, how similar one person is to another, or to other animals. Look at the map of the fruit fly. We're more similar than you might think.
It tells us how closely related we are to every other species on this planet. And it sets the human being for the first time in the context of all evolution of life on this planet. There's some wonderfully grand visions contained in that human genome sequence.
It's easy to imagine the grand ways the genome will change science and medicine. But first, some very hard work, turning all of those unrecognizable bits of information into something we can understand and then use.